Roy. I'm Dr. Rajesh Roy, the business head for micro division in MedGenome, which looks into the complete solution for infectious disease management. Today, MedGenome is the leader in all the applications of next generation sequencing. One of the applications of NGS is also to identify the accurate pathogen in sepsis, providing the best sepsis management for the patients. Our today's speaker, Dr. Gunisha, is a principal scientist for infectious disease and one of the eminent scientists in the field of NGS with years of experience in the field of microbiology and infectious disease. With that very, very short background, I request Dr. Gunisha to start her presentation on the topic, application of NGS in sepsis. Thank you, Dr. Gunisha. Thank you.
Yeah, thank you, Dr. Rajesh, for the introduction. So I think I'll straight away jump into the topic. Yeah, is my screen visible? And is the complete screen visible?
Yes, ma'am.
Okay, great. So I'll be talking to you about diagnostic value of next generation sequencing in sepsis. So before we just jump into how NGS, how next generation sequencing comes into sepsis, I just wanted to give you a brief background or introduction about the varied guest, viewers or webinars, listeners. They are from various backgrounds. So I thought I could just give a brief about sepsis, its diagnosis, the limitation of the diagnostic tools which we have now, and how NGS could be placed in all of this. So there have been many, prior to this, another two consensus definition of sepsis, but the third one is it is a life-threatening organ dysfunction caused by a dysregulated host response to infection. Now, when it comes to global burden, this is the 2017 data, which is published in Lancet in 2020, says that there are 49 million cases each year and there are 11 million deaths, which is pretty high. Now, when you see this number in children, then it is 20 million cases in children and 2.9 out of the 11 million deaths are in children. So this is a great concern. Sepsis is of great concern all across the globe. Now, when it comes to all these numbers, then you would see that among the low and middle income country, the burden is higher, which is 85%, and the deaths also are the similar numbers. And what is the Indian scenario? So we've seen this is a study which is published in 2017, and also I've referred to something which was published in 2020 in CHEST, says that the prevalence of sepsis in ICU is 56.4. Most common organisms obviously are bacteria followed by fungal, but here we should remember we are just talking about where the diagnosis, either it is clinical diagnosis or probably a blood culture is done. So you will never see viruses in all of these figures. Okay, so approximately 40% of the infections are multi-drug resistant, which is of a great concern. And the predominant source of this infection are either through pulmonary infections or abdominal or bloodstream as such, or a urosepsis. Now, among the neonatal infections, India has the highest incidence of neonatal sepsis, which is like 17,000 of the 1 lakh live birth, and the case fatality ranges from 25 to 65%, which is of again great concern. Now, if you see this slide, 80% of the sepsis death could be prevented if we have rapid diagnosis or appropriate treatment. Unfortunately, it's such a grave problem, but still the deaths happen. We do not have a proper solution for diagnosis of sepsis. Now, what are the different modalities, which are present? There are many, but I'm just listing out the principal ones which are actually used in various clinics and hospitals. First is, in many cases, is just the clinical diagnosis. There are various criteria guidelines to categorize this as sepsis or not. Of course, blood culture, which is a gold standard in many places, I mean, and many of the cases, blood biomarkers. In most cases, actually, it is done. And more recently, MALDI-TOF and molecular technique like multiplex PCRs are also used for sepsis diagnosis. Now, here I'm introducing next generation sequencing. I'll take you through how next generation sequencing could help in sepsis. I will take you through a literature review and also what is MedGenome doing about sepsis, because MedGenome core is into next generation sequencing and genomics. Now, as I mentioned, blood culture is the gold standard. So, and diagnosis is pivotal. If we have a diagnosis, definitely the survival or the mortality rate will not be high. So it's very crucial that early and adequate identification of the infecting species is there. So apart from knowing, the patient is having, it is very important to actually know the species of bacteria, fungi, or virus which is causing the infection, because the severity of the clinical manifestation depends upon the species of that microbe. And obviously, if we know what the diagnosis is, know the microbe which is causing the infection, appropriate therapy could be started. If we have all of that, we can control hospital outbreaks. Now, in case the negative blood culture comes in, does it exclude infection? No, because we know that the sensitivity of blood culture is very less. There are papers saying that it is 10 to 30%. There are few papers also mentioned 50 to 70, but the consensus has been like 10 to 30%, which is pretty low. So even if the negative blood culture is there, as empirical therapy is started, which leads to sometimes bacterial, antiviral, as well as antifungal therapies are also started, which also causes a lot of toxicity because of the antifungals. And obviously, if antibiotics are started, there's always risk of drug resistance in for all the bacterial infections. So as I mentioned, increased antibiotic usage is there, which in turn increases the development of antibiotic resistance. And in the case of neonates, it is a disruption of the nascent gut microbiome, which causes complete immune dysregulation. As you have seen in the earlier slide, the neonatal sepsis is so very predominant. Almost 1/5 of the deaths are in neonates because of the sepsis. Now, what are the limitations of blood cultures? As I mentioned earlier, the sensitivity is 10 to 30%. So they're false negative. Also, why there are false negative? Because there is usually a empirical antimicrobial therapy, which is already ongoing. There are microbes, fastidious pathogens, some anaerobes, and fungi, which do not grow as such. It is not, the blood culture does not support the growth of those organisms. And in children or neonates, sometimes, the required amount of volume which is required to be put in the blood culture bottles is not there. And so these are the reasons because of which the sensitivity is low. And also there is long time to, for the positive results to come. Though its sensitivity is low, the time taken for something results to come in for bacteria is 2 to 3 days. For fungi, it is 5 to 7 days. And for viruses, it does not support growth at all. Now, as I mentioned earlier, in sepsis is caused mostly in 50%, almost 50% of the cases from the pulmonary infection. And you know that in sepsis, there could be a secondary, it could be also obviously coming from the respiratory infections, and there could be a secondary viral infection caused by influenza and other adenovirus or so on and so forth, which goes undetected. There is no therapy given, or a empirical therapy has to be started. So here the TAT is not the issue. Here the issue is that the organism, the sensitivity is low and it goes undetected. Okay, this slide just tells you, most common bacteria which are isolated are gram-negative bacilli, which are isolated in blood cultures. Gram-positive cocci is 35%, and very few is yeast or fungi. And as I had earlier mentioned, viruses are and there are studies which mention that viral infections are in 1/3 of all the septic shock patients, and we do not even know about it. And there is no current recommendation for using empirical antiviral therapy in sepsis. So prudent, it is very important to actually screen for viral infection in this severe respiratory illness, which blood culture does not support at all. Now, coming to the biomarkers, there are various, I've listed them here: CRP, PCT, lactate, TNF alpha, but the utility is very much debated, across the number of hours, as you see, from sepsis to severe septic shock. And there is a lack of sensitivity and specificity, and always it's debatable, and there is a varying opinion when you do a literature survey. Now, apart from the ones mentioned here, also these specialized biomarkers are also used, but again, they lack sensitivity and specificity. So this slide basically tells you about the antimicrobial resistance. These are the common bacteria, gram-negative bacilli, which are isolated, and look at the amount of drug resistance which is there in all these pathogens. Like 82% of the Acinetobacter isolates have antimicrobial resistance, and similarly for Klebsiella and E. coli. So it's very important. It's a silent pandemic, and it has to be stopped. No, so I'll jump over to the molecular methods, establishing that low sensitivity and so much more should be done for sepsis patient, and how these molecular methods could help out. Now, RT-PCR based methods are there. There is a syndromic or multiplex RT-PCR based panels are there. I think Sanger sequencing does not help much. And in NGS method, next generation sequencing, I'll be taking you through the targeted and untargeted approach, and also giving you a brief algorithm which we follow at MedGenome more recently for clinical metagenomics and sepsis cases, where it involves initial PCR of pan bacteria and pan fungal PCR. So just taking you through what next generation sequencing is, genomics is it is driven by genomics, and next generation sequencing is massively parallel sequencing technology that offers ultra high throughput, scalability, and speed, and basically determines the order of nucleotides in entire genome or target regions of DNA and RNA. NGS has actually revolutionized the biological sciences, allowing labs to perform wide variety of applications, and more recently NGS has forayed into infectious disease, a segment too, and I'll be talking to you about it. So what are the basic steps? It involves DNA extraction directly from the clinical samples, and here we are talking about either whole blood or plasma sample in case of sepsis or bloodstream infection. The second step is library preparation. Basically, DNA fragmentation is done, adapters and indexes are put, not getting into technicalities of that. And then it goes into the sequencers, and the analysis is done, which is in case of sepsis, a proper bioinformatic line. The validation is absolutely important. I mean, I'll take you through. So I'm just taking you through what next generation sequencing is. Basically, the second generation sequencing is the next generation sequencing, and Illumina is the most popular. And in MedGenome, we have Illumina platforms, but more recently, for infectious diseases, and for metagenomics and clinical metagenomics, the third generation Oxford Nanopore or a PacBio, which is a long read sequencing chemistry sequencer, are also there. And we feel that future of genomics for microbes lies also among the third generation, that long read sequencing offered by Oxford Nanopore or PacBio. Okay, some misconception how next generation sequencing can be used. It is a very expensive technology. So this slide basically takes you through the cost of human genome, full genome sequencing from 2001 to 20, and we are in 2023, and it is completely dropped down further. The cost of whole genome sequencing is dropped to like close to $100, $150 or $200 as of now, which is extremely dropped down through the years, so that the conception that it is not affordable should not exist among all of us. So these are the workflow where, as I mentioned to you, nucleic acid extraction, DNA, RNA, or a total nucleic acid extraction, library prep sequencing, where quality control at each and every step is extremely important. Now, from next generation sequencing, I would want to take you further ahead and tell you what metagenomics is, because metagenomics approach is the one which it is explored in literature, and we are at MedGenome also exploring a metagenomic approach. So I just wanted to take you through. So metagenomics is powered by next generation sequencing. We need to use those high throughput sequencers to actually sequence. So what is metagenomics? So this is basically study of structure and function of entire nucleic acid isolated and analyzed from all the organisms. Okay, and to study a specific community of microorganisms such as residing in a gut, human skin, soil, water, air. And in this case, we are talking about a clinical sample, we are talking about whole blood, we are talking about plasma. It is a culture independent genomic analysis and genomics. So what is the difference between genomics and metagenomics? So genomics explores complete genetic information of a single organism only. But when it comes to metagenomics, it explores a mixture of DNA from a multiple organism in a community. Now here we take example of blood extraction, total nucleic acid extraction is done, and which obviously is a mixture of human DNA because it is from human. It will also include microbes, and if there are bacteria, there are viruses, fungi, bacteriophages, all will be sequenced all together. So this basically this picture depicts that various matrices, it is all mixed, mixed community, DNA is extracted, it goes through a metagenomic workflow, which I mentioned. The workflow of next generation sequencing remains the same, and then there is detection of the pathogen. Now, taking a step further from metagenomics is there are two different approaches for metagenomics. One is an amplicon sequencing, or it could be a metagenomic sequencing. Amplicon sequencing, I would say it is more targeted approach, and in metagenomics, it is completely untargeted kind of a approach. So because I'm going to in detail talk about what amplicon sequencing is and metagenomics, so I thought I will just give the two concepts for the metagenomic approach. Okay, so this slide basically tells you, the use of or clinical metagenomics. How does it go? Again, amplicon sequencing and metagenomic approach. There's a clinical sample. The DNA extraction is done. The DNA would include, if present, obviously human, and if present, bacteria, fungi, parasite. Everything will be sequenced. We have to have a separate approach for bacteria, fungi, parasite, and this is the untargeted approach where it will be a mixture of everything. Now, in case if you're looking for bacteria only, if there is a suspect that only I want to look for a bacteria, then we use a 16S RNA gene-based primer for amplification of bacteria. If you're looking for fungi, it is an ITS-based amplification. Directly, PCRs are done. That's why I mentioned a pan-fungal and a pan-bacterial PCR, which I'll introduce you here. So PCRs are done from the sample which is extracted. Now, if there is a pan bacteria positive, then it is taken through for sequencing, and if pan fungi is positive, it is taken through the next step of next generation sequencing. And if both are positive, it is taken through. So here I will talk about the algorithm which we at present we follow, and how we want to improvise based on the demand of the market, and so on and so forth. So this is the amplicon, where a separate bacteria, fungi, parasite approaches, PCRs, have to be done and taken through sequencing, while metagenomic is as such, where RNA, DNA is extracted. The RNA could be reverse transcribed into cDNA. Now, here in the case of amplicons, since we are doing PCR, it is taken through library prep, and then we understand what kind of bacteria, fungi, or whatever is present. While in this case, there are two approaches again in metagenomic approach. We go ahead with cDNA and DNA conversion, and then again we have a hybridization protocol. Basically, if we want to target certain organisms, certain bacteria or certain viruses, then probes could be added. So something what we do here is we follow a metagenomic approach, and then we have a hybridization step involved where we have a panel for Candida, basically an invasive Candida infection, which will help in two things. We have probes for all the Candida species. Apart from identifying the species of Candida which is present, it also will tell you about the drug resistance genes which are present in fungi. So that's the power of metagenomics. That even in that we can use a targeted approach. Other approach is obviously an untargeted metagenomic approach where all the nucleic acid is sequenced. Here in this case, targeted only few, or like I'm giving you example of Candida is what we're doing, and we can go ahead with that. So these are two approaches. So I'll just take you through a literature review and talk about metagenomics, how it has been helpful in sepsis. I'm quoting some of the latest papers here. And so what MNGS is metagenomic next generation sequencing. So in all, this paper had higher positivity rate than culture, especially for bacteria, viruses, and multi-pathogen infection. As you can see here in case of blood, the culture, it was just 59 which were positive. This is a small study of around 100 odd samples, and 90 of them were positive by metagenomics. And this was by Chen et al. And published in 2020. This is another paper in BMC infectious disease in 2021, again, which talks about, comparison between culture and metagenomics, where this is a larger study of 300 samples, where only 27 samples were positive by culture, while a big number of 144 samples were positive by metagenomics. So this is also done blood and even in bowel sample, and this also talks about paper also talks about its use in CSF. So clinical metagenomics is to look forward to. So apart from blood at MedGenome, we also look at metagenomics of all in non-sterile, sterile site samples like CSF, peritoneal fluids, or in case of bowel, blood, any of these samples. So metagenomics is a tremendous technology which is actually revolutionizing the field of infectious disease. So this is another paper, 2018. Here, instead of culture, they had used a clinical diagnosis as the benchmark to compare the metagenomics, where then they found that sensitivity was 95.16 and specificity was low. So they do talk about that, NGS reports must be carefully interpreted to avoid, because usually the NGS based report, it is usually polymicrobial. I'll take you through why it could be polymicrobial, because NGS is very sensitive technique and will pick up all the microbes while collecting blood, or the microbes present in the kits and the centrifuge tube, and how algorithms are required, cutoffs are required, guidelines are actually needed to define how these reports could be actually generated and interpreted. So this paper also talks about subjective judgment of clinician is still needed, which is greatly influenced actually by the clinical experience also. So there is not all hunky-dory about metagenomics. There are also limitations. I'm going to talk about those too. But this is fantastic in terms of comparing, where gold standard is giving a sensitivity of 10 to 30%, and something like even 95% or specificity of 76 is a worthwhile thing to go ahead with. So this is another study. So there are studies where they have done total nucleic acid extraction. They've gone ahead with the DNA extraction, and then to cover the spectrum of all the microbes present, you can go with total nucleic acid, convert the RNA in that to cDNA, so that RNA viruses, DNA viruses, parasites, fungi, and bacteria could be included. There's another thought process where which is actually gaining a lot of interest, and also a commercially available solutions are available for sepsis, is where the DNA extraction is the cell-free DNA extraction is done. cell-free concept is very popular in case of cancer genomics for actually understanding if those biomarkers are present and cancer-causing biomarkers are there. But in terms of infectious disease, also cell-free DNA, there are specialized kits which are available for doing cell-free DNA extraction, and obviously the workflow of the NGS remains the same. However, it is very important, again, as I mentioned, to have controls and as well as the patient. So each patient sample has to be run with the control, either a non-infected control or the controls which are added from the laboratory sides, and then understanding what are present microbes present in the non-infected or the non-template control, and what is actually present in the patient's sample. So obviously, bioinformatics input is needed, algorithms are needed, and after that, expert evaluation by the clinical microbiologist, from the ID specialist, and also from the clinician is also required in such cases. Here, if you see the results, again, in this particular study using cfDNA, cell-free DNA, a comparison is done where SIQ basically is a sepsis interpreting quotient, which has been shown here. It is 73%, it gave diagnosis, while blood cultures mere 13%, with a stark difference again. So in literature, there are multiple interpretation criteria because it is becoming more and more popular. There are a lot of interpretation criteria given by different authors. I've not put all of them. I've just one for you here, but it undergoes debate, and we also have our own interpretation criteria based on our experiences. This I'm just quoting that in case if it is a metagenomics. Results come in if it is Mycobacterium species or Legionella pneumophilia are seen, even if the number of sequencing read is 1 per reads per million, then it becomes important. Similar in the case of, but significantly different from human, from adenovirus and influenza viruses also. And when it comes to fungi, greater than 5, or in parasite greater than 10 reads per million are used as a positive criteria for a species. But there are various criterias, interpretation criterias. Unfortunately, there are no proper guidelines established, but yes, a lot of interpretations criteria have been mentioned. So giving you background of, I've taken you through the different diagnosis modalities. I have taken you through next generation sequencing. I've introduced you to metagenomics and the two criterions, the whole genome metagenomic or the untargeted approach, and as well as the amplicon based approach. So I'm just taking you through our experiences, what we do here and what we offer. We offer a whole genomic approach where a comprehensive sequencing of all the genes, including human and organism, is something which is done at MedGenome. We also do an amplicon-based approach using 16S rDNA among the various variable regions of 16S rDNA. V3, V4 is the most popular. Obviously, somebody is looking for eukaryotes, which is none. I think for sepsis, I think we should just stick to 16S. And obviously, for interpretation, there are SILVA databases. Now, when it comes to fungi, as I mentioned, ITS1 is the most popular. It can identify if in a pan-fungal PCR, it would come positive for fungi. And then we'll take it through the metagenomic analysis and find out if it is fungus, yeast, or mold. Again, negative controls are extremely important to doing a background negation and also establishing cutoffs. I mentioned to you earlier, there are also hybridization-based approach. We have a Canada panel for identification of various species, and we're having excellent results. We are doing a study also regarding this with our another hospital in west of India. So apart from actually identifying the Canada species in the plasma sample, we have curated a database based on where there is no database which is available as such, apart from the MARDI database, which again needs to be updated for the antimicrobial resistance. We have also curated from various peer-reviewed journals and created a database for finding out what kind of antimicrobial resistance is there. So this is the diagnostic algorithm for now, which we do for difficult cases in sepsis, as well as other clinical metagenomics, other clinical samples which are difficult to diagnose when it comes to CSF infections, endocarditis cases, and also in prosthetic joint infections. This is very, very popular among the clinicians. As the day we see multifold increase in the interest of doctors, because none of the other modalities, other diagnostic modalities, give them answers. So that is why they are leaning towards this approach, which we offer for now. Is again, since I mentioned targeted, amplicon based, and metagenomics. So the algorithm is we extract the DNA. We do a pan-bacterial and a pan-fungal PCR, and which either of them come positive, we take it through metagenomics and generate report. And similarly, if in one shot, if a clinician wants to know that the patient has bacteria, fungi, or viruses, a virus infection, then metagenomic approach obviously is the method for them. This is just giving you an example of some of the reports. So it is usually polymicrobial, where cutoffs have to be established. Some inputs has to be done from your ID specialist. It also has to be from input from to interpret the results. Now, in it comes to this, this is also a semi-quantitative kind of method. Semi-quantitative in the terms based on the sequencing reads which we generate, we can give a semi-quantitative idea as to what abundance of the organism is present in your blood sample. Now, if you come to this, the Serratia species is like 26.48 abundant. There is Corynebacterium, Staphylococcus, and Propionibacterium. Now, based on our experience of a microbiologist or ID specialist, the interpretation would be, again, it is subjective, but yes, that is why we give a research use only report, wherein we know that all of other species could be contaminant on the species, and Serratia is a gram-negative bacteria and could be the causative organism for that particular patient. Now, similarly, for fungi, for the pan-fungal PCR, if it comes positive, this was one of the report. We also, since there are a lot of fungal spores present, and especially in India, then running a negative control is extremely important to cut off the background, while in this case there was no reads in the negative control. Here they were. So when we look at this, there were some reads also present in the negative control. So we do not consider aspergillus and leptodontium here, but something which is not present in the negative control but present in the clinical sample becomes very, very important. So it is not yes, no kind of a situation like we are used to in PCR or blood culture, positive, negative. There is some input which is definitely required to interpret the result, where both clinicians as well as the patient needs to be aware that this is the kind of report which is going to be shared. But obviously we are working on the algorithms here to the subjective factor which is there, which needs to be seen manually, could be taken off, and it could be included in a bioinformatics pipeline and algorithm. So all that is again taken care of, and more of clinician friendly kind of report can be given. However, each and every report is discussed with the ID specialist as well as the treating doctor in this case. So these are the options which are for sepsis, metagenomics or NGS based options which we have with us. But I also just, since we're talking about sepsis and this is the sepsis week, sepsis month, sorry, I think it was on September 13th, and that is why we are all meeting up and MedGenome's initiative to actually talk about sepsis. I just wanted to mention that again, need of the hour is directly, having some diagnosis directly from the clinical samples. There's some options of multiplex which are available in market, where actually you have to do a blood culture, and then positive blood culture is taken through multiplex PCR. But again, as I mentioned, the sensitivity of culture is low. So how that helps is it's very subjective and actually debatable. But there are and which have been actually used in many hospitals. There are newer techniques, multi-top, but again, they are culture dependent. Again, comes a problem. So, bacterium coexist in approximately 20% of the cases. So, with the fungal infections, this I think I missed mentioning that. So, it's very important to have bacteria, fungi, viruses all looked upon at same time, which becomes very, very important. So, even for multiplex PCR, it is various things are very critical, especially when we're doing the DNA extractions. Are we taking care? Fungal have different cell walls, gram-positive are different to crack. So we take care of all of these modalities. Extraction are done well, internal control, amplification controls, negative control being run, and positive control run with each and every sample, which helps in interpretation of the results. And a thorough validations of sensitivity and specificity. And the panel which I'm talking about is a CE-IVD approved panel, which we actually offer. So this is the sepsis panel, which has the 5 Candida species. It has 9 antimicrobial resistance, covering vancomycin. It is covering MECA, and it is also covering all the carbapenems and all of those for the gram-negative bacteria, and bacterial agents, which include gram-positive, and the most common bacteria which are known to cause sepsis. Again, this does not cover viruses. Now, if there is a respiratory infection, then probably a respiratory panel has to be taken. So in all of this, NGS-based approach, again, definitely we cannot say that this isn't good, but something which you're offering, but definitely a NGS approach is more wholesome kind of a approach. So it has why it is good, because it is unbiased sampling, broad identification of known, unexpected, novel pathogens. I mentioned to you a quantitative or a semi-quantitative data could be given based on the sequencing reads, and quantification becomes very, very important, especially most of the reports are polymicrobial, which I just showed to you. That the one which is most abundant, which have the proper cutoffs, should be considered, and some of them should not be. However, there are a lot of challenges, challenges for metagenomics. I thought I should mention that too, because NGS is a very sensitive technique. It may produce false positive, that is why cutoffs are important, controls are very important. So these are challenges, but there are solutions to it also. So all the ketome, labome can be negated by including a negative control, where the background obtained organisms obtained in these kits and labs could be negated from the list of microbes which have been listed out from the clinical sample. Background biodata is crucial for interpreting. This is what I was been talking about. Cutoffs are very, very important. Interpretation criteria are very important. Many of them are subjective, but based on the experiences, it has to be established. In cases of polymicrobial infections, predominant pathogens are detected, and less abundant pathogens may be filtered out, where high detection threshold should be done. Host depletion becomes very, very important, because if you're extracting from plasma, 95% or 99% of the sequencing reads will be of the human. So we're doing extraction, we're removing the host reads initially, and then which increases the sensitivity of the metagenomics. Also becomes very, very important to have a standardized bioinformatics pipeline, extremely crucial, which will decrease of or remove the subjectiveness or the more involvement, manual involvement of the laboratory staff or the ID specialists or the clinicians. So interpretation, again, very important, and there are actually unified diagnostic criteria are unavailable right now. So this slide basically tells you all about what I mentioned, the challenges: human host background, the contamination, host depletion, then there is contamination here, obviously cost, but as mentioned, the costs are coming down. Turnaround times is something which are higher for now, but we were working towards making it less than 24 hours. Because that's most crucial for sepsis patient. Computational powers are definitely needed. Bioinformatics, software validation, and criteria cutoffs are very important for all of this. And obviously, guidelines have to be there from the clinical microbiology or sequencing board actually to help out in using this. For now, this is available as a research use only. So this slide, I think, just talks about the power of NGS and microbial genomics. This is not something about sepsis, but I thought I could just talk about that. More and more there's interest in microbial genomics, where apart from whole genome sequencing of microbes, interest in metagenomics, which I talked to you in detail. Clinical metagenomics is what I was talking to you about, and there's a lot of projects which we do for ecology and environmental studies, microbiome studies, drug resistance studies, for all the whole genome sequencing of microbiome, sequencing of plasmids, bacteriophages, microbial transcriptome, metatranscriptome, and dual RNA-seq. And we feel that Oxford Nanopore generation sequences future of microbial genomics to reduce cost, reduce stat. It will be an excellent solution. And these are just the genomic platforms which we have. Most of them are Illumina here, and we also have Oxford Nanopore and other platforms. I think we have to update this one. Also available with us. So thank you with this, and I'll be happy to take any questions from the audience regarding this. Yeah. If there are, yeah.
Dr. Gunisha, there are a couple of questions on the chat box. If you can just check the chat box.
Can you help me with those? Because I'm not able to see any messages here. Rajesh or Dheeraj?
Yeah,
one of the question is secondary infections also take place post-infection. Does metagenomic NGS helps in predicting the possibility of secondary infection?
Yeah, see, in case if microbe is present in the clinical sample, metagenomics will be able to pick it up. So as I mentioned, secondary infection caused by viruses or something, it is basically just feeding in the DNA and interpreting each and every nucleotide out of it using algorithms. So if it cannot predict, obviously, that it can just tell if it's there. Yes, it will be able to tell you that apart from bacterial infection, the secondary fungal or viral infection is present. Yes.
There is a second question from again. What are the cell-free assays based on metagenomics NGS? If samples are taken from patients, what is the type of sample?
So it is basically whole blood, which is taken in something called specialized tube called Streck tubes, which are freely available. And blood could be collected in those Streck tubes and transported. And
no, I think what is the type of sample? So
the whole blood in Streck tubes. These are commercially
in case of
available.
sepsis. Yeah.
In case of sepsis, in Streck tubes. For cell-free DNA extraction, the blood has to be collected in Streck tubes.
Next question is, what are the diseases other than sepsis which requires metagenomic NGS analysis?
Okay, so basically clinical metagenomics has brought any sterile site sample can be used. As I mentioned, we do a lot of projects we are already doing for blood, plasma, CSF, prosthetic joint infections, synovial fluids, endocarditis samples, bowel samples. So this could be a solution for any sterile site. a sample. It could help. The algorithm remains the same. The workflow remains the same for all of them, and it is actually very popular among among the neurologists, and especially the prosthetic joint infection orthopedicians. Yes.
Next question is turnaround time.
Okay, the turnaround time for now is slightly on the higher side. We aim to give reports in 7 to 10 days. Then you would say how it's helping the blood culture, but I mentioned to you blood culture sensitivities are low. So it is 10 to 30, even if it is 7 to 10 days now, it would definitely help the patient, but we are working towards actually bringing down less than 24 hours. And I think probably next webinar we could have, when we have that, I could show you the results where we are actually doing it less than 24 hours, hoping for that soon. Yeah.
And I think it also gives a picture of the secondary infections
Yeah, absolutely.
also. Yeah.
Yes. Yeah.
Yes. Next question is, you had mentioned a little bit about Candida panel. Why do you have a separate panel for Candida? Could you please elaborate on that little bit?
Oh, this is actually a project we are doing for a client where Candida infections, I mean, were the most, they were studying those Candida infections. So we developed a pan-Candida panel for them. And it turns out it's been pretty successful in giving us good results, and also gives us insight, that similar panels could be launched. But this is a specialized project which we are doing, and we are actually going to expand this into Pan Canada to a pan fungal kind of a panel, where we add more of clinically or medically relevant fungal species into the panel, and like Aspergillus mucor and Rhizopus and other species into the panel, and make it as a pan fungal panel, or a fungal sepsis panel. So this is just one step towards it. And a specialized group or tertiary care center working on Candida infection had requested for us, and we see a lot of success, and gives us more confidence into offering this as a pan fungal soon.
Next question, presently, which technique is better?
Sorry, for sepsis?
Yes.
Okay, so presently the options available or are obviously limited. There is blood culture, there is sepsis and metagenomics. I'm sure with all these if somebody the clinicians are okay with the research use report, I would recommend to explore a metagenomics option, and then only the clinician will have more confidence into it, because it gives a complete wholesome view. So I think metagenomics is a good option right now, if the criteria is not the clinical report.
I think there is a part in the question. Presently, which technique is better, nanopore or Illumina?
Okay. So I think it's, I said future is Oxford Nanopore, obviously, because it's shorter turnaround time and more cost effective. Yes. For clinical metagenomics, Oxford Nanopore is something which is good. But Illumina, our core isn't to Illumina, and there are solutions also available which we're exploring with both the platforms. But for now, I would say that yes, Oxford Nanopore, when it comes to TAT, it is a better option for now.
Next question, can the metagenomics NGS give a readout of immune cell types near the site of infection?
Okay, we are not doing, we are looking completely into microbial, but if you want to look at the host, then it's a different game altogether. A whole exome or that kind of a scenario can be done. But here, if you're looking at metatranscriptomic approach, probably all that could be answered, or a dual RNA-seq. But if you're looking just for diagnosis per se, sepsis, metagenomics is good. But if you're looking at host response and the transcriptomics, so there are different modalities and in a research mode. Then we could discuss that project. That could be discussed in person, yes.
Next question, what are the bioinformatics software or tools can be used in metagenomics, helps in analyzing the sample?
Oh, there are a lot of tools. I cannot enumerate them now. And there are in-house pipelines which are proprietary and we cannot disclose many of them.
How long it take, next question, how long it take for sequencing and validating the sample?
The sequencing the
Metagenomics.
sample. Okay, the sequencing, as I mentioned, we aim at giving a TAT of 7 days. This is because our sequences run on particular days. But when it comes to just giving out results on a Illumina platform, the results could be given out in 2 to 3 days also. The working times are pretty less. The lab time is few hours, I would consider.
The next question is the current turnaround time 7 days, as you have mentioned, due to the bioinformatics pipeline? Which step of the process is the bottleneck?
Okay, here bottleneck I would see is running on a bigger platform. Is the Illumina platform which we are running on. Not a bottleneck, I would say, but we are working towards using Illumina platform within a day. So currently we have 7 to 10 days, but using both ONT as well as the Illumina platform, it will be brought down to less than 24 hours. This is presently we working on all different modalities and all the challenges which are there in metagenomics to get hold of all of them, have criteria, have bioinformatics pipeline validation. So challenges will be negated. What we have is very, very temporary. And using both platform, our aim is to get it below 24 hours. It can be done using both. Yeah.
The same guest's question is, does the nanopore have any disadvantages due to the long reads? Does it affect the sensitivity?
Okay, there are always the Q30 values for the long reads are always lesser than the Illumina. Definitely, that's the. But we're not here. We're looking at microbial identification. We're not doing any mutation analysis. So I don't think so. The disadvantage of the long read or its lower Q30 values will be a challenge in identification of organisms.
Next question, what other panels would you consider interesting besides Candida?
I mentioned to you a pan-viral or a pan-fungal panel will be an excellent, or a pan-pathogen is something very exciting, and we've been working on that. Or a sepsis panel as such, a targeted sepsis panel. There is a metagenomic approach, which I mentioned about it, there is the untargeted approach, but there are also like a Candida panel. We could have a pan pathogen where a wide thousands organism probes could be mixed together, and or sepsis panel or a pan bacterial or a pan fungal panel. All that is something which we are working on. Yeah.
I think we have got these questions so far.
Okay. I think very interesting questions, and I think the audience have been listening. I'm thankful for that, and I think soon, I'm hoping in few months we'll be doing another webinar where I could give you more specifics as to how the TAT could be reduced and what kind of platform. But yes, both the platforms have their own advantages and disadvantages, but both offer something very nice and unique for sepsis, and which is coming up. So we are very excited at MedGenome, especially we've been working, trying to deliver something for our clinicians for sepsis, finding solutions for sepsis for a long time. I think our first offering is for now is the targeted approach, higher TAT, but extremely useful, extreme good feedbacks which we've been getting from clinicians. And obviously we have a multiplex panel which we could offer for now. Yeah.
And
But
I think one interesting aspect of it is for immunocompromised patients, especially for the onco patients, the cancer patients or transplant patients. This is playing a very important role. What is your point into it, Dr.
yeah,
Gunisha?
absolutely, definitely. I
Because there's a
agree
lot of
with this.
chances of co-infections.
Right, absolutely. Most of them are polymicrobial infections also in transplant and other immunocompromised. So this will be definitely of help. Yeah. Yeah, it's just that when we have the right algorithms and bioinformatics pipeline and good criteria, and also from the society assets, microbial society or molecular biological assets, we have good guidelines if they are published and validation across different labs and state. That's also a very nice thing in future to have guidelines actually uh, from all these institutes or from the bodies who actually govern all of this diagnostics into microbes.
So great. Thank you, Dr. Gunisha. I don't see any other questions in the chat box. It's 8 p.m. and we are on time. So thank
Right.
you for, thank you all the attendees for attending this webinar. And we are very, very hopeful to come up with some other exciting talks very soon on metagenomics. Thank
Right.
you.
Yeah. Thank you, everyone.
Thank you. Bye-bye.